Hello, welcome to another video from the conscientious biologist Ben Gallagher. This is cell 7, it's that extra one that I've had to cut microscopy into. It looks at electron microscopes and magnification. Hopefully you've come straight to this one from the cell 6 light microscopes and resolution one. But if not, a very quick reminder that resolution is about the clarity of a picture. It's about how small you're seeing an actual object, so that when you magnify that up, you can still see all those tiny little details. Images taken with low resolution never captured the tiny details, so magnifying them won't make any difference. So what we're looking at now is electron microscopes, and we're looking at how the resolution of those is so much better and what those can produce. This is still part of the GCSE course, so it follows on directly from cell 6, as I said. If you enjoy it, if it's useful, please do subscribe to my channel if you haven't done so already. Thank you, good luck. So if we want to capture at high resolution the internal contents of a cell and try and capture things like mitochondria and chloroplasts and ribosomes, we need a type of microscope that can give much better resolution. And that's these things, electron microscopes. Now, electron microscopes are very different, okay? They're massive for a start. You look at this one here, the actual electron microscope there I'm putting in a box for you. But you'll see it's mounted up on a desk. That desk is a really special desk. It's not just like any table. It's got special pads on the bottom. It would have to be mounted on a floor that's like vibration resistant because the microscope can't vibrate at all. It's hooked up to loads and loads of computer power and software because it's a really high-tech piece of kit. The electron microscope uses beams of electrons instead of light. Now, by using beams of electrons instead of light waves, you can give really high resolution, really high resolving power. That's because electrons move at a much faster, much shorter wavelength than light waves, which means they can see much smaller things because they get interfered with and bashed by much smaller objects. But there's some advantages and disadvantages to that. Okay, but the major advantage is when you magnify images taken with an electron microscope, you get lots of detail of very small things. So if you want to look at anything really microscopic in massive detail, you've got to have one of these things. However, there's pros and cons to this, and you do need to know these advantages and disadvantages. This table comes up a lot in the exams, or it might ask you, it might be a 3 or 4 mark question, give some advantages and disadvantages, or compare an electron microscope with a light microscope. So the advantages are huge. If you look at the advantages column, there's only 2 things in it, but they're massive. Electron microscopes give very high resolution, which means you can have very high magnification because you can keep making it look bigger and bigger, and it will retain the detail because of that high resolution. Disadvantages, though, they're ridiculously expensive. A light microscope that you might have in school, you might be able to get a reasonably decent one for kind of 60 or 70 pounds. Light microscopes, we're talking thousands and thousands of pounds. Really good ones, we're talking millions of pounds. So they're very expensive. So there's very few schools that would have an electron microscope. I only know one school that has an electron microscope of its own. Most universities do, but universities obviously have the funding to have them. Another big disadvantage of an electron microscope is you can only view dead material. So under an electron microscope, you could put living cells and you could literally watch the cells do mitosis. You could put tiny living creatures like Daphnia, little water fleas under them, and you can watch them swimming about and interact. You can watch their little hearts beating through the light microscope. You can't do that with an electron microscope because that chamber marked in yellow has to be in a vacuum. So all the air gets sucked out, so nothing in there can survive, nothing in there can live. So you can only view dead material. They have to be kept and used in special controlled rooms. You have to control the humidity, control the temperature. They're very delicate pieces of equipment. So it takes a special expensive room to even house them in. And lastly, they're not portable. You look at the size of it. Okay, a light microscope you can pick up, you can put it in the cupboard, you can get it back out of the cupboard, you can take it to a different room, you could take it out in the field with you, take it out into the woods if you want to look at things. An electron microscope just isn't portable. You can't move it around like that. So its advantages are enormous, but it's not all good. There are some disadvantages of an electron microscope, which means there are advantages of a light microscope. But if you want really clear images, you've got to have an electron microscope. There's two main types of electron microscope. The first one is a scanning electron microscope. That scans the surface of objects. So if you want to look at a solid surface and get a perfect image of it, that's the one you want. But they don't have as high a resolution as the second type, which is a transmission electron microscope. That allows you to see through objects. So if you're putting a thin slice of cells on there, you can look through it, which means you can see into the object that you're looking at. So transmission electron microscopes have a much higher resolution. I mean, there's transmission electron microscopes now that allow you to see atoms. They're that good. They can magnify things up to 2 million times. That's pretty amazing. So there's two types, advantages and disadvantages in both, but you do need to know about electron microscopes and what they're for. Now, we did this with light microscopes. We looked at some images, so let's look at some images from an electron microscope as well. And let's pull this up. Have a look at it. Any guesses as to what it is? That's a mitochondria. Okay. Now think about how small they were when we showed the diagram of that cell in the first slide. They're ridiculously small. They're about 2 micrometers long. But look at the detail that's been revealed in this. When I drew my mitochondria, you might remember I put little sort of wobbly lines coming in. Well, look how long and thin those things are. They're called cristae. But you can see all those tiny little internal structures. You can see now the size of the membrane that's there. They actually have a double membrane, which is why it looks slightly darker. But look how small you can see. And also look closer to where I am. You see those long lines that look kind of white, but can you see they're covered in black dots? Well, those black dots are ribosomes. And we said ribosomes were like 30 nanometers, but they're revealed in this diagram. Not in great detail. You can get electron microscopes that can look at a ribosome and show it in massive detail. But on this diagram, you can see they're just tiny little black blobs. But those are absolutely ridiculously small things. Let's pull this up. That is a loop of DNA. It's a circular chromosome from a prokaryote. So now we're talking at something down on a molecular scale. So this has been highly, highly magnified by an electron microscope with very, very high resolution to see the detail of something that small. This was taken by a by a scanning electron microscope now. So you can see we're not looking into cells here. We've scanned the surface of them. But this is a bacillus bacteria, those blue things, bacillus bacteria. They don't actually look that color. What you can do with electron microscopes is once you've got the image, you can use computer programs to color it as well, to make them look a bit more visually impressive. It's false color, so they're called false color electron micrographs, and a micrograph is a picture you get through a microscope. But it's still really cool. Think how small bacteria are about the same size as mitochondria. They're about 1 to 2 micrometers. So you think how small they are, and look at the detail of its surface of that cell. That's pretty amazing. But what's really cool, you don't just have to look at cells. I love this picture. This is the face of a mosquito. So think how tiny mosquitoes are, those little buzzing, annoying things that can sting you. This is its face, and look at the detail on that face. Look at the tiny hairs. Look at all the tiny little bits of its eye. Look at its mouthpiece, its proboscis. It's amazing the detail that we've got on that picture. And again, that would have been taken through a scanning electron microscope because it's a scan of the surface. It's not looking through that. But some amazing, amazing images can be got through electron microscopes. So last key thing, and this is just a bit of a skill, but it does seem to come up every single year in the exams, is calculating the size of an object. You've got to know how to get magnification, and you've got to know how to manipulate that equation. So I'll walk you through this slowly, but it's reasonably straightforward. So magnification is the observed size of an image divided by its actual size. So whatever you observe, whatever you're looking at in an image, say it's a photograph, if you divide that by the actual size of the thing you're looking at, you'll get the magnification. Okay, we'll go through an example to explain what I mean there. So for example, let's pull up that picture of that mitochondria that we had a minute ago. Now we said on the first slides how big these things were. So you know the actual size of a mitochondria is 2 micrometers. So we've got the actual size that could go into our equation. What we need is the observed size. What size is it that you observe in the image of the mitochondria. You sometimes hear the observed size referred to as the image size. It doesn't matter which one you use. It's the observed size of the image. Okay, so how can you work out the observed size? Well, that's really obvious. You just put a ruler on it and you measure it. If this is the diagram, then I put my ruler along it, I measure it, and I go, right, well, the observed size is 9.3 centimeters because I've just measured it. Now, obviously, I don't know what size screen you're viewing this on, so that 15 centimeter ruler may not be 15 centimeters on yours, but it doesn't matter. At the scale, I've got it on my screen here, that's 9.3 centimeters. Okay, so there we go. I've got everything I need. Observed size divided by actual, 9.3 divided by 2. No, and don't fall into that trap. Okay, it's not 9.3 divided by 2 because the units are different. First thing you need to do when you're making this calculation, you must get them both to the same units. Okay, easiest way to do that is take the biggest one and convert that one. So 9.3 centimeters is 93 millimeters because they're 10 millimeters per centimeter, and we know 1 millimeter is 1,000 microns, so it's 93,000 microns. Now they're at the same level, they're both in microns, so now we can do our sum, we can put them in. It's 93,000 divided by 2 is 46,500 times magnification. So that picture of the mitochondria is at 46,500 times magnification. It's 46,500 times bigger than an actual mitochondria. Okay, so that should be pretty obvious as to how you do that. But let's just make sure you can manipulate that as well. So hopefully you're quite familiar with these mathematical triangles for use on equations. If I just divide that triangle up and I put those key things in. So I'm going to put magnification, and we know magnification is observed over actual. So I can put that in, observed at the top over actual. So if I want magnification, I can see that observed is above actual. It's acting, that line going across is acting as a division. I've put a division sign in there for you. Observed divided by actual gives me magnification. But what if I didn't want magnification? What if I wanted was the actual size? Well, if I wanted the actual size, then I just look to what I've got on the other two. It's still observed at the top, and now that's observed divided by magnification. So if I observed something that's 10 centimeters, and I know that's been magnified 40 times, then it's 10 divided by 40. That would give me the size of the image. Okay. What if I wanted the observed size? What if I knew the magnification, I knew the actual size, but I didn't know how big the image was? Well, that line going down is like a multiplication. So that would be magnification times actual would give you the observed. So whichever one you want, you cover up, and you just look at what the sum is for the other two. It's a simple little manipulation triangle that I'm sure you've seen in maths if you've been working out speed for distance divided by time or things like that. Okay, it's an easy way to use it. And you can abbreviate it to just this, the MOA, magnification observed actual, MOA. Okay, it's an easy way to have it on screen so you can manipulate that. So if you need to know the actual size of an object, you can measure the image and you can just divide that by the magnification. That's the most common way it comes up in the exams. They'll give you a picture and they'll say, here's a picture, this is a 150 times magnification, how big was it in reality? You get your ruler out, you measure it to get the observed size, convert that to microns or nanons if it's something really small, micrometers and nanometers, sorry, and divide that by the magnification. It's pretty straightforward. Okay, hopefully you'll be absolutely fine with doing that because it's not that tricky. So key points and summary of this presentation. It's a pretty easy one. There's not a lot of detail here. Microscopes allow us to see microscopic objects that aren't visible to the naked eye. Microscopes magnify objects to make them big enough to see. That's basically all they do. Magnification is just how many times bigger an object is in an image. But resolution's the key thing, because resolution is how detailed and clear an image is. And resolving power is how well a microscope can distinguish between things. That's the kind of official definition of resolution. It's the ability to distinguish between two objects. Okay, it's basically you're trying to resolve the gap in between two things. Do they look like one? Or if you look really, really closely, is there a tiny gap that you can resolve to tell that they're different things? It just means you can tell what small things are there. Okay, and there's no point magnifying something if the resolution isn't good. Okay, so a microscope with high resolving power can produce high resolution images, which can be magnified to show high levels of detail, even in very, very small objects. You saw that in that mitochondria, which was only 2 micrometers long. Electron microscopes have much better resolving power than light microscopes, but they're really, really expensive, difficult to use and to move around. And lastly, you need to know that equation, that magnification is the observed size divided by actual size, and just try and remember the triangle so you can manipulate that equation. But there's not really a lot in this unit, and that's the end of the cells unit. So you can now move on. This completes the cell biology unit. I suggest next, and if you've gone back to the first slide with the big 5, the 5 questions that simplify biology, that give you the full playlist, this is what I've suggested you go to next. This makes the most logical sense and will help build your learning. Go to the transport through membranes presentations. The first one of those is diffusion and active transport, and then that goes through osmosis and examples of osmosis. Head to those now. Please do subscribe to my channel if you haven't done so already. And if you've enjoyed this video, click the like button so I know what kind of things work and what doesn't. Thanks a lot. Good luck with the next section.